west in any case. All right, so our first speaker this afternoon, or are we in the afternoon? Not quite, we're not Pacific time in any case, is Maria Elizabeth Baeva, is that right, Elizabeth? Maria?
Yes.
Good, thanks, I should have practiced. All right, and she is working in the lab not ready. Okay, yeah, no, of Wilf Jeffries, want to make sure. All right, so Maria Elizabeth, I'm going to put your, I'm going to share screen and we'll get rolling.
Sounds good.
That good?
Perfect.
Okay, go for it.
Okay, so hi everyone. First of all, thank you to the organizers of Norman Bethune for allowing me to present today. And today I'm going to be giving a short story about the link between ocular pathology and Alzheimer's disease. Next slide.
Oops.
Okay, so I'm sure most of you are familiar with Alzheimer's disease. It's a neurological disease that's characterized by cognitive impairment, typically in the elderly, and that usually is caused by brain atrophy. And we think what's causing this brain atrophy is the accumulation of a protein called amyloid. However, despite the fact that we've known about this disease for over 100 years, there's been very little clinical progress in terms of therapeutics available. One of the big reasons for this is that it's actually really hard to see what's going on in the brain of live patients without using very cumbersome, expensive, and labor-intensive equipment. One of the symptoms that our lab has previously shown to occur in Alzheimer's patients and in mouse models is that in addition to this amyloid accumulation, there's this increased presence of leaky blood vessels. But that still doesn't address the fundamental problem of, well, how do we see what's going on in the brain? But it's not just the brain that seems to be affected by Alzheimer's, it's also the eyes. So the retinal microvasculature of the eyes of Alzheimer's patients has also been noticed as being altered compared to age-matched non-Alzheimer's controls. Next slide. And so we hypothesize that the same pathological hallmarks that we see in the brains of Alzheimer's patients and mice, including accumulation of amyloid, increased angiogenesis, and the breakdown of blood barriers, are actually also pathological hallmarks in the eyes as well. And so we use this mouse model, the TG2576 mouse, which expresses human amyloid precursor protein with the Swedish mutation under the promotion of the hamster prion promoter. And so what this does is this creates a lot of amyloid expressed primarily in neurons, but also in immune cells. Next slide. And so what we see in figure A is, as expected in the brains of these mice, there is a lot more amyloid. What's interesting is in figure B, we see the eyes also have a lot more amyloid as well. And if we take a look at figure C, hopefully you can all see that. This is a cross-section of the retina of a TG2576 mouse, is that this amyloid is accumulating in very specific spots of the retina. And this actually correlates with where blood vessels are located in the retina. So there seems to be this perivascular clustering of the amyloid. Next slide. When we look further into the vasculature of the retina, what we see is that it's not just that these blood vessels, that there's more blood vessels, but that these blood vessels are dysfunctional. So when we measure CD105, which is a marker for angiogenesis or leaky blood vessels, like in figure A, we see that TG mice have more of this marker in their eyes. And we know that these blood vessels are dysfunctional because in these mice, like in figure B, we see there is much more albumin. If the retinal blood barrier integrity was maintained, we should not be seeing more albumin in the eyes of these mice. And so in figure C, we have another cross-section of the retina. In green, we are showing CD31, which is a marker of vessels. And if we take a look at the choroid, which is on the right-hand side of the retina, unfortunately, I didn't have space to put a comparison between wild type, but trust me on this one, there is more vessels in the choroid of the TG mice compared to the wild type. Next slide. So all that being said, this is really good support for using non-invasive ocular techniques, something that you would find at your eye doctor, to see maybe if we can use the eyes as a proxy for what's going on in the brain. Of course, there's going to need to be more investigations. For example, we're going to need to quantify and qualify the changes in the vasculature in living mice. These were all done ex vivo. And we're going to really need to strongly correlate the changes in the brain with the changes in the eyes of these mouse models as well. Thank you so much for paying attention and for listening. Thank you to my lab, and I'll be happy to answer any questions.
That was great. Do we have any questions? Well, I have questions. So, so there are other diseases that are associated particularly with aging that are going to happen concurrently with Alzheimer's disease, like age-related macular degeneration, diabetic retinopathies, hypertension. Are you going to evaluate mice? If in the human condition, that will probably complicate matters. How do you deal with all those factors?
That is actually a great question, and that's probably another reason why there's been such a lack of therapeutic options for Alzheimer's, because as we age, everything seems to go wrong all at once. What's interesting about all those diseases that you mentioned is that vasculature is something that seems to be affected in all of those cases. And this actually does kind of support the idea of maybe some of those medications or some of the therapeutics available for those diseases could be used for Alzheimer's as well, because even if they are related even slightly, if they have overlapping pathological hallmarks, maybe targeting one of those can help in some way. I hope that helps answer your question.
Yes, it is. Okay, and Helen Cotier has a question about, have you looked at whether amyloid accumulates in other tissues than brain and the eye vessels?
That's a great question. I've actually been very recently looking at platelets in particular, but we're also interested in other tissues that are separated by some kind of blood barrier. So we're actually thinking of investigating testes in the mice as well.
Cool. Okay, thank you. Thank you very much, Maria. That was excellent. All right, so we'll move on to our next speaker. Is Olga Mykhailova from the lab of Dr. Jason Acker at the University of Alberta and the Canadian Blood Services. Olga, give me a second because I'm doing that.
Sure.
All right.
Thank you for the introduction. I saved a couple of seconds. I would like to start with a little bit of background and the study design.
Okay, hold on. I lost your slide presentation. Hang on.
Sure.
There we go. And
so today I'm going to talk about one of our recent studies that shows that donor sex affect the hypothermic storage lesion in subpopulations of young and old red blood cells. I would like to start with a little bit of background and the study design outlined on this slide. So in healthy adults, billions of red blood cells are produced per day and released into the peripheral blood where circulate for about 120 days. Thus, at the time of donation, there is a heterogeneous distribution of red blood cells from recently matured young to senescent old red blood cells. However, for those who donate frequently or for young females who are menstruating, this distribution could display a higher proportion of young red blood cells due to increased erythroid. So we hypothesize that young female frequent donors may have a high ratio of young red blood cells, which are less susceptible to the storage lesion. This could also explain the effect of donor variability on the storage lesion of red cell concentrates. The objective of this study was to determine if the donor sex can affect the quality of different red blood cell subpopulations throughout hypothermic storage. To do this, red cell concentrates from healthy donors, 4 females and 6 males, were percolfractionated into the less dense, young, and denser old red blood cell subpopulations and stored for 28 days with weekly quality assessments for key red blood cell parameters. As we move on to the results, firstly, we assessed red blood cell indices and their morphology. As you can see in the top right corner, concentrates of young cells had a disc to discoid red blood cell shape, while old consisted of mostly crenated discoids and spheroids. The difference in their structural properties was also confirmed by lower MCHC and higher MCV in young cell concentrate compared to old ones. Furthermore, according to the mixed model analysis shown in the table, MCV and therefore hemoglobin autofluorescence parameters are affected by donor sex, with both being higher for females. The next slide outlines the hemolysis and deformability of red blood cells. As we expected, hemolysis levels linked to the donor sex with lower values for females. Moreover, the rate of changes of hemolysis, given in callout, was many times higher in old red blood cells of male donors than that for other subpopulations. They have also assessed the mechanical properties of the cells, as seen from the rigidity chart in the bottom right corner. Paired comparison shows a significant difference between female and male donors in different groups at different time points, but according to the mixed model analysis, there was no donor sex effect. Thank you. No donor sex effect, which is probably due to the small sample size. Okay, move on to our next results of metabolite accumulation, and shown at the bottom, metabolomics analysis also indicated a significant sex-dependent signature. Regarding subpopulations, both markers of increased oxidant stress, glutathione disulfide, and decreased capacity to cope with these as adenosylmethionine were detected in concentrates of old cells. On contrary, shown at the top, imaging flow cytometry assessment of reactive oxygen species showed their high content in young cell concentrates right after the separation, but by day 28, their content was on the control level. Unlike old cells, which seem to have failed to mount a good enough antioxidant defense. According to the mixed model analysis, donor sex has an effect on both intercellular metabolites, ROS and calcium, which were higher for female donors. So, summarizing all the above on the next slide, we can draw several conclusions. Storage lesion of red blood cells can be defined by red blood cell heterogeneity after donation. Donor sex affects the physical, structural, and functional properties of red blood cells. And last but not the least, young red blood cells of female donors are less susceptible to the storage lesion and age slower than old red blood cells of males. I would like to thank CBS for providing me with a postdoctoral fellowship, all CBS donors, and all the members of Dr. Jason Acker Lab who made this work possible. Thank you.
Thank you, Olga. That was great. Do we have some questions? Well, I'll ask. Why is this happening? Why do you think that there's a difference between female donors and male donors? What teleological?
Let's say the most obvious answer here is going to be increased erythropoiesis. If we are talking about healthy donors, so we can get those increased erythropoiesis just from the female donors who are menstruating, or for donors who are donated their blood frequently to just enhance this erythropoiesis also.
And are your numbers big enough to exclude other factors other than sex?
Oh, no.
There may be other reasons.
I'm afraid not. For this particular small study, we were more concentrated to see if there is some difference which is linked to the donor sex. So it was just like a first start of our big study where we're going to have lots of different groups with different number of donation, sex, and maybe any other, let's say, smokers or not smokers, pre-donation hemoglobin level, and so on.
Great. Okay, good. Well, thank you very much, Olga. All right.
Thank you.
Okay, so our next speaker is Wenhui Li, also from the lab of Dr. Jason Acker, the University of Alberta and the Canadian Blood Services. Wenhui, you're going to run this, or am I running this? Your mic is on. Ask to undo your mic in a second. We made her a co-host. Yes. Oh, you're doing it.
With that, you can see that.
That's perfect. You're right into your first slide, except that you're not on, there you go.
Okay. Yeah.
Okay.
Thanks for the introduction. So my topic today is the role of donor sex on the percentage of CD71 erythroid cells in whole blood. So the CD71 erythroid cells are type immature red blood cells with the expression of CD71 and CD235A. It also, the maturation process also shows Olga's presentation. It's the same as that one. So the different mature red blood cells present in bone marrow. And the expression of the CD71 increases firstly, and then decreases when the red cell getting matured, especially in bone marrow. And also it accompanied with the CD235A expression, which is a glycophorin. And when the red cells get going out of the bone marrow, it also get rid of the nucleus, and then went to the peripheral blood, which also is the young red cells I would call. So what is the CD71 then? The CD71 is a type transferrin receptor which mostly present on the surface of the red cells. Here as is the green one shows. So it mediates the iron transferrin into the cells with the help of the iron reductases and with the help transported to form hemoglobin, which is the oxygen delivery. It's really important for the criteria to be 105. Further, those are actually or blood give the blood their hemoglobin actually lower than the criteria. That might be testing error or hemoglobin fluctuation. And then further study to show that to test the ferritin level, which can show how much iron in their body. It shows that some donors are actually in the risk of iron deficiency. Here it shows that specifically for the frequent female donors, there are 27% actually in iron absent, and even 66% are iron deficient, in the state of iron deficient erythropoiesis. And for males, that will be 60% and 48% respectively. And so this shows that female donors have a higher risk of the iron deficiency than males. And how the hemoglobin relates to the CD71 then. A study for the patients with the cancer, with anemic shows that the hemoglobin level is actually correlated with the CD71 cells. Here shows that the hemoglobin level in the horizontal shows that hemoglobin level increases and the CD71 decrease actually. And also an important subpopulation of the CD71 cells also shows a similar trend for the hemoglobin and the CD71. So this brings us a scientific question: is there a higher percentage of CD71 cells associated with the anemic donors? And to be further, whether there is a differences between the male and the female. And hopefully we will do some study about how the CD71 cells would reflect donor health. So we developed a flow cytometry method to determine the CD71 cells after antibody titration and voltage titration and all the things. We did a preliminary data for the CD71 frequency in whole blood. There are 2 young female donors and 1 young male donors. We tested for 2 days. It shows that the CD71 cells from both the young female are higher than the young male both in these 2 days. Well, of course, this is not enough to prove that actually the female has a higher frequency of the CD71 cells than the male. So we are going to collect more data to investigate the CD71 cells between male and female. And we are also going to test how this would reflect the donor health. Yeah, thanks. Thank you for that.
Hi, Wenhui. I have a question actually, if you can
Yes.
hear me.
Yes.
Yeah, I just want to ask you have you already looked at these CD71 during the storage period, if that's going to be also affected? Because according to your slide at the very beginning, that you see more of the CD71 at the beginning, at the early stages in the bone marrow, but then decreases when they go to the peripheral blood. But after you collected and stored your blood, you see also a high number or high expression of the CD71 on the red blood cell during the storage. Is that differ from early storage versus old storage?
You mean the results showing that at the slides, like it was in the like we collect the blood and we tested immediately, like within 24 days, and so we did it 2 days for that. So it is in the blood with the EDTA.
Okay, and how many samples? You said 2 females versus 1 male when you already collected the differences between them, or is that like just like a showcase? Have you really looked in farther sample size?
No, we are going to do that. But like I went back to China, so like we stopped. So we are going to collect more data later on. But this is just a very preliminary data for the CD71 cells for now.
Sure, thank you.
Nope. Thanks.
Okay, thank you very much. All right, and we will move on to our next speaker. So Dr. Michelle Zeller, or Mickey Zeller, is an associate professor and clinical hematologist in the Division of Hematology and Thromboembolism at McMaster University and the co-director of operations transfusion medicine for Hamilton Health Sciences. She is the McMaster University program director for transfusion medicine, a focused competence a diploma program at McMaster, and a medical officer at the Canadian Blood Services. Prior to joining the faculty at McMaster, she completed a postgrad training in internal medicine and hematology and transfusion medicine, as well as a master's in health professions education. She is the director of education for McMaster Center for Transfusion Research, where she leads and participates in clinical research on blood alternatives, including intravenous iron, blood product utilization, and informed consent, and impact of blood donor characteristics on recipient outcomes. Nikki, it is all yours. Can't hear you though.
Can you hear me now?
Now I can. All right, you're on, and we can see your slides. Perfect.
Okay.
Yeah.
Great. And can you just see the one slide as opposed to presenter view?
I can just see the one slide. It's like perfect.
Perfect. Awesome. Good afternoon. Here it's more into the afternoon. I'm in Hamilton than when you guys are. I'm sorry that I didn't get to come in person to Vancouver in April, but this is fun too. So thank you to the organizers for inviting me to speak about donor characteristic impact on recipient outcomes. These are my disclosures, and I would especially like to thank and recognize Professor Nancy Teitel and Dr. Jason Acker for their support and encouragement as I've been expanding within this field, and their contributions to content in this talk, as well as recognizing MCTR and CVS for their ongoing support. Today, I'm going to begin this talk with a review of proposed underlying biologic mechanisms that support differences in red blood cells depending on donor age and sex. And you guys have heard from other people who are engaged in this field and making other really exciting findings. So you can add this to what you've heard previously. And then we're going to move on to a discussion in the literature on clinical outcomes that look at donor age and sex impact on recipient outcomes specifically. Mortality in patients transfused in hospital is probably somewhere around 8 to 9%, and that's what was demonstrated in other large clinical trials in transfusion medicine. And I often wonder to myself, what if there was something beyond ABO and RHD that we should be considering when we're selecting an ideal unit for transfusion that could maybe impact this mortality rate, decrease it even just a little bit? Sex mismatched transfusion and transplantation have been associated with poor outcomes in other areas of transfusion and transplantation. For example, plasma from female donors, we know, is associated with increased risk of transfusion-related acute lung injury or TRALI. Literature looking at sex mismatched heart transplantation has demonstrated an association with increased transplant-associated mortality when you get a sex mismatched heart. And stem cell transplants from female donors have been demonstrated to be associated with worse outcomes, while younger male donors appear to be associated with better overall outcomes. So I'm left wondering, should donor sex and age matter for red cell transfusions also? Donor age can affect red cell quality, as you've heard from previous speakers today. And some of the areas in which age we know impacts red cell quality include reduced erythropoiesis, which could lead to reduced hemoglobin and red cell counts as people age, increased mutations, enhanced red cell aggregation, and changes to erythrocyte membrane and fluidity, which can adversely affect cellular deformability. As donors age, there's decreased ability for bone marrow to generate new red cells, and the average biologic age of circulating red cells increases with donor age. In addition, we see changes to immune tolerance, inflammation, and oncogenicity as we age. Biochemical and biophysiological differences between male and female red cells have also been well documented, and again, as you've heard earlier today, and go beyond just changes in hematocrit, cell volume, hemolytic propensity, and deformability and mean cell hemoglobin content. For example, females are more likely to produce antibody-mediated cellular activation by donor antigranulocyte or anti-HLA antibodies. And male donor units, as I mentioned, have higher hematocrit and total hemoglobin. Male donor-derived products also have higher grades of hemolysis, regardless of differences in processing method, and there's a higher testosterone level in men. In addition, donor sex, particularly during the reproductive years, results in significant differences in the age distribution and physical characteristics of young and old red cells in circulation. For example, progesterone-treated red cells will exhibit reduced storage and osmotic hemolysis, as well as improved adenosine triphosphate maintenance. The impact that differences in red cells and subpopulations of red cells have in different donor demographics is an emerging field, as you've heard. Let's now talk specifically about how these changes might translate into harm, and we'll start with from male red cells transfused into female recipients. Transfusion of biologically older red cells from male donors with high hemolytic propensity, lower deformability, and higher densities may impact oxygen delivery, coagulation, and vasculopathy when transfused into a female recipient. Larger amounts of hemoglobin, particularly free hemoglobin that are found in male red cell products, could overwhelm the haptoglobin scavenging capacity of the RES, or reticular endothelial system, in female recipients, and this could result in reduced nitric oxide bioavailability, and that can result in endothelial dysfunction, platelet aggregation, and oxidative injury. In addition, hemodynamic changes in blood flow due to the transfusion of male red cells, which have a higher ratio of older, denser, and less deformable cells, could affect the margination of red cells in circulation of female recipients, which subsequently can impact platelet and white blood cell interactions with the endothelial system. And how about why female red cells may be harmful for males? While the characteristics of red blood cell subpopulations in male and female donors may have different immunologic characteristics. For example, subpopulations of erythroid precursors in young females and pregnant women are enriched with immunosuppressive cells, and these have been shown to impair pathogen defense in neonates and cancer patients. What follows from all of this, the last number of slides that I've shown you, is do these differences in blood from older and younger people, blood from males and females, translate into a risk of morbidity and mortality to recipient outcomes in vivo? So what happens when we take this into the clinical field? And that's what I'm going to focus the next part of this talk on. This first study, an observational study on donor age impact that I will present, is a Canadian study through the Ottawa group, where they looked at 30,000 plus patients who were transfused between 2006 and 2013. They used donor age as the primary exposure of interest, and the secondary exposure was donor sex. The primary study outcome they were interested in was recipient survival. They show that risk of death statistically was significantly higher for recipients of red blood cell transfusions from younger donors. The second study that I'm presenting on this slide is a cohort study with data on blood donors and transfused patients that were extracted from SCANDAT, or the Scandinavian Donations and Transfusion Database. In this study, they simplified their analysis differently than the first study I presented, and they included only the first 7 days of transfusion for each person. And their analysis showed no association with overall 30-day and 1-year mortality when they looked at association for donor age specifically. The same Canadian study of 30,000 plus patients from the previous slide also looked, as I mentioned, at their secondary exposure at recipient sex. So this next slide is looking at observational studies that looked at donor sex impact. And this first study showed that regardless of recipient sex, each additional unit of red cells from a female donor was associated with an 8% increased risk of death compared with exposure to a male red cell. The second retrospective study that I'm presenting on this slide of 31,000 plus patients from the Netherlands concluded that exposures to red cells from an ever-pregnant female donor was associated with increased all-cause mortality among male recipients, but there was no increased risk for female recipients. These studies are both in contrast to another study from the SCANDAT database, which included patients from Sweden and Denmark who received at least one red cell transfusion of allogeneic blood over a decade's worth of study, so from 2003 to 2012. And they found that donor sex was not associated with patient mortality. So we have some conflicting findings here. And that was one of the reasons that the McMaster Center for Transfusion Research group, led by Professor Nancy Teitel, led a study to investigate locally the association between blood donor sex and age and transfusion recipient mortality by analyzing our local donor data, our local transfusion data that we linked to donor data spanning a 6-year period. And this study included just over 25,000 transfusion recipients receiving almost 100,000 red cell units. Using a careful analysis that controlled for covariates and stratified on time-dependent and fixed variables, a significant association between donor sex age, and recipient outcomes was found. It's kind of difficult to see, so I'm going to summarize on the next slide. So as I mentioned, both donor age and sex were associated with increased risk of in-hospital mortality, and there was a significant increased risk of mortality for female recipients who received male red cells. Both male and female recipients transfused with red cells from younger donors, that being less than 45. And there was a similar trend, though not significant, when male recipients received red cells, when male recipients received female red cells. We also performed an analysis on the impact of both age and sex in the same model, as opposed to splitting the model. And here we showed that receiving sex mismatched red cells and red cells from donors less than 45 years were both found to be associated with an increased risk of in-hospital mortality. The fact that there are a number of observational studies and different exploratory analyses with conflicting data led to us to decide to conduct a meta-analysis and systematic review. And our goal was to summarize evidence examining the impact of sex mismatched red cell transfusions on recipient mortality specifically. We searched in our meta-analysis and systematic review for both randomized controlled trials and observational studies. And we looked at overall certainty of evidence for each outcome of interest using the grading of recommendations assessment, development, and evaluation system, or grade system. We found 5 retrospective observational studies with just over 86,000 patients represented, and there were no RCTs that we found that met our specific inclusion criteria. Sex mismatched red cell transfusions were associated with a higher risk of death compared with sex matched transfusion. Similar results were found when all follow-up durations in each study were considered, and that's what's shown in this study. So we applied a 3-level meta-analysis to account for differences and different weights of the exposure periods, and we found a very similar finding. And to summarize these findings, again, we found increased mortality demonstrated in recipients of sex mismatched red cell transfusions compared to sex matched red cell transfusions, and the hazard ratio was 1.13. And we had a similar hazard ratio of 1.1 when we considered all follow-up durations. The overall grade certainty for the outcomes of mortality was very low. This pooled estimate was based on observational data, and therefore our certainty started at low, and it was subsequently lowered to very low because of issues related to risk of bias with our included trials. As I mentioned, all of the trials, or all of the studies, were observational. And therefore, when I present these results, I'm very clear to state that these findings cannot be interpreted as evidence of a causal relationship because observational studies are at higher risk of bias. The 3 potential sources of bias that we felt were most contributing were the confounding by indication, co-interventions like transfusion of platelets and plasma, and then classification of exposures as well, which impact risk of bias in many transfusion-related studies. I wanted to step back and just think about what can we learn from the Age of Blood saga? What can we take from those decades of study and resources that went into Age of Blood? Do we need to iron out a biologic mechanism and repeat observational studies until there's consensus before pursuing prospective controlled clinical trials? I mean, I think the answer is no, because observational studies will always be so Ottawa, mortality associated with improved survival compared with female-only red cells. in all hospital patients, and their target is 8,000 patients recruiting. And I'm told recruiting should be wrapping up pretty soon, maybe in the next 2. They'll be meeting their targets, and look forward to learning results soon. A proposed study that I have been trying to put forward is a little bit different than the ITADS question. I think we'll put some complementary data if we do this first pilot study. Our question is, in transfused adult patients admitted to the intensive care unit, and secondary outcomes will be a better patient. That important. There is in the literature, and that the evidence derived from observational studies we have in time, and that there are yeah,
We're having some problems. I don't know what to think your end. So we're getting some problems.
well, I'm asking. I can switch to speaker on my phone if that helps at all.
Now it seems to be okay. I wonder if that was it's solved now.
Is there a part that I should repeat? Was it wonky throughout?
No, it's just wonky for the last 4 slides. Do you want to go back just a little bit?
Sure, to the lessons learned from Age of Blood.
You talked about the lessons learned from Age Blood saga, and then you started to talk about the how should we go forward in terms of.
Okay, so I'm so sorry about that. I thought that my headphone would be the best way to prevent any audio glitches.
Yeah.
So the way I think to move forward is we don't have to have ironed out biological mechanisms that are 100% explaining the observation, because what we can learn from the Age of Blood saga is they had over 30 observational studies that were conducted. They had decades of research that looked for in vitro explanations for potential changes, but after there were well-conducted RCTs, these demonstrated that fresh red cells were not superior to stored red cells in the majority of populations that were that have been reported on. And so I think it's safe to say that we should move forward at this point with some well-designed clinical trials to help further elucidate a potential relationship between donor age, sex, and recipient outcomes. And I'm aware of one trial that is in progress at this time, an innovative trial assessing donor sex and recipient mortality, or ITADS, which is being led by Dr. Dean Ferguson and Alan Tinmouth in Ottawa. And their question is specifically, is transfusion of male-only red cells associated with improved survival compared with female-only red cells in all hospitalized patients? And their target for recruitment is 8,000 patients. And I'm told that over the next month or two, they might have completed their target recruitment of 8,000 patients, which is very exciting. So I am very excited to await results of this study. And I'm hoping as well that I will be able to lead a study on a question that's slightly different than ITAD's. In transfused adult patients admitted to the intensive care unit, is the risk of in-hospital mortality lower when donor-recipient sex-matched red cells are transfused compared to sex-mismatched red blood cell transfusions? And again, this would be specific in ICU. We'd be focused on our primary outcome of in-hospital mortality with a number of secondary patient-important health outcomes, and we're proposing first a pilot for feasibility. So to summarize, there is evidence in the literature to support donor characteristic impact on recipient outcomes. However, that evidence is conflicting. The evidence is derived from observational studies which lend to bias, and therefore we cannot make definitive associations with the studies that we have. And furthermore, as you heard from talks earlier from today and also from mine, there is a lot of work going into exploring biologic mechanisms that may explain potential differences. But finally, there is need for rigorous prospective clinical trials. So I apologize again for any audio challenges, and I would be pleased to answer any questions or try to answer any questions. And thank you again for having me.
That was great. Unmute. Thanks, Mickey. That was fine. Okay, you can't have a symposium like this without some technical glitches. While I'm sure there will be questions, but while we're waiting, I've unmuted, or I've allowed people to unmute themselves as well, and chat is open. When these observational studies, when when bloods or red cells are selected for use in the hospital, are the red cells labeled male, female? From a male donor versus?
So right now, no product that comes out of Canadian Blood Services is going to be labeled. We can assume that the plasma in the freezers in the blood banks are male plasma, and that the plasma that platelets are suspended in is male plasma, but the red cells are not labeled. Okay. However, we've been able to work with Canadian Blood Services, and Ottawa group has as well, in order to retrospectively look at what the sex was of the donor who contributed a transfusion and link that to recipients. And for the ITAD study, Ottawa worked with Canadian Blood Services to affix labels where the clinical side was not aware of what they were getting, but that way the blood bank could know what they were sending out to do this prospective study. And that's something that we would certainly be considering as well.
No, I was just curious if there's bias, potentially bias at that stage, but there wasn't. So that's interesting. Are there other questions? Oh yeah, there are. Do these studies track irradiated status of red cell units?
I don't believe that the Canadian, so I'm not aware that they commented on irradiated status of red cells in the studies that I've looked at. I know that we don't in Canada irradiate all of our red cells, and I'm not aware of any center in Canada that irradiates all of our red cells. However, I am aware in the States there are some centers that will irradiate everything. Most commented on, I think they all commented on leukoreduction, but I don't believe that each individual unit commented on irradiated status or that was included in analyses.
Other questions? You can unmic, you can speak on the microphones, or you can chat. No.
Hi, Mikey. Thanks for this excellent presentation. Actually, I enjoyed it. I have one question because you also mentioned in one of your slides that the increased risk of either like in-hospital mortality or the death increase with the number of transfusion. But one of my question is there other factor could also contribute to the increase of the hospital mortality or even like the death, such as like not also the age of the donor, but also maybe the age of the blood cell, the number, of like different sex matches or any other factors?
So the studies that were in the systematic review that I looked at, they handled the confounders in different ways, and most provided a list of all the different things that were controlled for. And for example, 4 of the studies accounted for whether people got other types of transfused products or not.
Right.
The analyses varied considerably, and I think after having gone through the motion of really, deeply thinking about the different studies and the different conclusions that they made, I was really left with this profound feeling of the analyses. The difference in analysis could have accounted for the changes that we're seeing, and that's why we need prospective studies. So if an analysis can change the conclusion that you make, to me that's very scary, and that means we need to, look at something else. Now, each of the studies had very good reason and scientific rigor for why they included or excluded or controlled for or didn't control for, or how they controlled for some of these different confounders. But they all did it differently. I'm not sure if that answers your question fully.
You already did. And okay. And I have another question, if I'm allowed to.
Go ahead.
Like you already mentioned also, like because of the loss of biological and biochemical differences between the 2 groups, male versus female, that could also contribute to the mechanism of why we would see such a kind of impact post-transfusion. However, do you have like a main mechanism that may be the central reason for this impact?
So you asked me this, and also the grant agencies that I applied to for funding asked the same question. Let's have one focus. Is there one thing that you can pick and focus on to make this just a little simpler to wrap your head around? And my answer is no. There isn't one thing. We were kind of banking on maybe this immunologic differences in male versus female might be supporting some of the observations that we're seeing. But how quickly would you see the changes? How far out could these impacts be? Some studies use a 30-day follow-up period. Some use 12 years. Would the receipt of one transfusion, in a male of a female bag of blood in 12 years, does that make sense that would cause an impact on their mortality 12 years later? So I have not yet read something that sort of gives me there's one thing that is leading to the harm from male to female and female to male, and you can believe that I have pestered Jason Acker for his thoughts on this as well, and maybe somebody else on the call right now, that has that magic one that they think explains this all. But for me, it's so complex. This is so complex, and I just wonder, is it enough for us to think about ABO and RH when we're dealing with such complexities in the bag and from the donor variability and the heterogeneity of the recipients it's going into? Is there more that we need to consider to make sure the optimal unit from the optimal donor is going to the optimal recipient? But I can't think of just one thing. I don't know if anybody else on the call wants to chime in.
No, that's worth a huge grant. Nobody's going to tell you.
Thank you.
All right, Mickey, thank you very much. That was wonderful. Really appreciate you doing that.
Okay, thanks. And I apologize again for the feedback there on the audio. Thank you again for having me, and take care.
Thank you, you too. All right, so we'll move on to our next speaker. This is without slides. This is Mr. Robert Cooper, is a multi-award winning writer, director, executive producer, who's been involved in the creation of over 400 episodes of television. Now you might wonder, why do we have this kind of speaker? In any case, his latest series is called Unspeakable, which aired in 2019 on CBC and Sundance TV. It chronicled the Canadian tainted blood tragedy and was produced through his production company, Meso Entertainment Inc. And it's actually the rationale, as you know, for the creation of the Center for Blood Research is as a result of the tainted blood tragedy. Mr. Cooper is also known for having produced and co-created the record-breaking Stargate television franchise for MGM. So Rob, thank you very much. And he's on the screen. He's not got.
Hi everyone.
So you can tune in and.
Can you hear me? Sounds good.
Yes, it's fine.
Great. Hello. Thank you for including me here today. Let me know if the sound cuts out at all.
No, you're good. You're good.
Okay, good. As Ed said, my name's Rob Cooper. I'm 51 years old and I have hemophilia factor VIII deficiency. I've been very lucky over the course of my life for a number of reasons. I have truly amazing parents, a wonderful, supportive, understanding wife who might be listening, and 3 incredible children. But there are a couple of other big things that led me to be here today talking to you in reasonably good health. The first is that I was born not long after cryoprecipitate was invented. Prior to that, life expectancy and quality of life for hemophiliacs was, to put it simply, not good. I do have a couple of fused ankles, but for the most part, I'm doing okay. Beyond what I've mentioned, the big reason I'm lucky is what I'm here to talk about. As you probably know, concentrated factor products began being used in the early 1980s in Canada. This was around the time HIV and hepatitis C were infecting the blood system. I was in my early teens. The new revolutionary concentrates were being pooled from thousands of donors, which obviously greatly increased your risk of exposure to contaminants like a virus. Just take a second and imagine going to a gathering right now with thousands of people packed into a small space, no masks allowed. Add in a mortality rate for those infected of more than 60%. That's the party blood product users were being invited to back then, in most cases with no knowledge of the danger. Early on, there wasn't much out there about the mysterious virus later named HIV. My parents, however, were extremely cautious people and sought out as much information as they could. Critically, they also met someone named Bill Mindell, who I'll talk a little bit more about later. My parents asked my doctor to keep me on cryoprecipitate, which they hoped would be a little less risky than concentrates because of far less exposure to the number of donors. When he said no, they moved me from the Hospital for Sick Children's program in Toronto to the adult program at St. Michael's, where I was very well cared for by Dr. Jerry Teitel for many years. I was very happy to see he's speaking right after this. My story and many others play out in a somewhat fictionalized way in Unspeakable. It first aired in 2019, and if you haven't seen it yet, I hope you will check it out. For the second life-altering time, I was extremely lucky. So many others like me were not. I managed to avoid getting HIV. I did get the consolation prize, hepatitis C, and lived with that for more than 30 years. I went through 2 very long, difficult treatments that failed, and then in 2014, as my health was beginning to decline, the 3rd treatment finally worked. I was cured, or at least from a clinical standpoint, I could say the virus was rendered undetectable. That was when I first started thinking about my experience as a storyteller instead of as a victim. It became clear to me that I was in a unique position to pay tribute to other people who could no longer speak for themselves, people who fought for a safer system, compensation, and justice. In most cases, doing so while they were sick and dying. I pitched CBC the idea of making a show about tainted blood, and that began the greatest challenge of my professional career. How could I possibly do justice to such a huge and painful story that affected so many? The first thing I did was option 2 books, Vic Parsons' Bad Blood, The Unspeakable Truth, and Andre Picard's Gift of Death. Both men are journalists who covered the story. Vic is also the father of a co-infected hemophiliac. My team and I spent more than a year doing research, interviewing dozens of people, and I quickly realized that even though I had lived through it, I actually knew a lot less than I thought. Up until the recent COVID pandemic, tainted blood was the biggest medical tragedy in Canada's history. There are many similarities and many differences we could unpack. How many COVID deaths could have been prevented? Could we have been more prepared? Could we have acted more quickly? You could certainly argue the utter fiasco of long-term care in this country contributed, but we're still in the midst of it. In the case of tainted blood, we have the lens of time to look back through. Many things shocked me during our research. Maybe the most surprising was that so many people today had only a casual awareness of what happened, or worse, they had no idea the blood scandal even happened at all. People who actually lived through it, well, we heard more stories than we knew what to do with, and it was quite overwhelming. One of the common questions you get asked when first pitching a television series to a network, especially when it's a period drama, is how is it going to be relevant to today's audiences? I'm obviously biased, but even before COVID, the relevance couldn't be more obvious. If we have handled this current pandemic well at all, then I believe it is at least in part because the tainted blood inquiry and the subsequent battle for victim compensation brought about accountability and reform that changed Canada. Our biggest challenge, besides finding a way to cram decades of tragedy, turmoil, and human resilience into 8 hours of television, was trying to understand why it happened. Motivation is very important when making a television drama. Why did those bad decisions get made? What were people thinking? The federal inquiry led by Justice Horace Creever went a long way to answering some of those questions. I was lucky a few times. The side note, it was quite thrilling when he wrote to say how much he liked the show after it aired. I've been told that any praise at all from him is very rare and should be cherished. The fact is, the most satisfying thing about making the show was hearing from many affected survivors and families of victims how much they appreciated their stories being told. But back to the inquiry, which illustrated the systemic breakdown. Systems are complicated and often impersonal. You could say it was the institutions responsible for public safety that made decisions that led to mortality. But when it comes down to it, people made those decisions. As we have seen of late, new viruses are tricky and not easy to understand, and it's reasonable to say there was much we didn't know back then. For a time. Ultimately, there came a point where vital information was withheld, and the sad fact is, tainted blood was knowingly distributed to users. Certainly on an individual level, there were doctors and healthcare providers across the country that made what turned out to be the right choices. Doctors like Manchu Poon and Christos Soukas stood out from the crowd. The inquiry identified a system that had little checks and balances and placed the most important decisions in the hands of people whose primary concern was ultimately not what it should have been, and that's public health. Some said the inquiry went too far, others said it didn't go far enough. The mandate given to Justice Creever prevented him from pointing to criminal intent. Certainly money played a huge part. Some of the internal documents I got my hands on were downright infuriating in the way they explicitly put a price tag on people's lives. But it was much more than just bureaucrats trying to save a few dollars. What Creever ultimately did was make recommendations for how to make the system better, and I do think we've all benefited from that. Ultimately, the subsequent criminal charges and trials failed to produce the kind of justice victims had hoped for. Nothing could undo what had been done, but the hope was that accountability might prevent it from happening again. Ultimately, billions of dollars in compensation have been paid to victims and families. In some cases, it made things a little easier to survive, but nothing could replace what had been lost. Further to the issue of relevance to today is the way the stress of the COVID pandemic has shone a light on much needed light on other systemic failings. Some systems are actually built to keep people down. Even those built with good intentions can end up imperfect. All of us need to recognize that when they are flawed and work to change them. And to do that, we need both the will and the resources. As we see in minute-by-minute social media updates, politics gets in the way because it's short-sighted, greedy, and often dishonest. Back when AIDS emerged, there was no internet, no Facebook or Twitter, no cell phones, no way of instantly communicating information or rallying large groups of people behind a cause. One of the leaders in the show is inspired by Bill Mindell. I mentioned him earlier. He was one of the heroes of the real story, father of a hemophiliac, a whistleblower, consumer advocate who eventually helped get safe blood products into the hands of consumers. There's a scene early in the show where the character based on him is asking a provincial minister for help. It's suggested the complaint will be addressed at the next quarterly meeting of the appropriate committee. Imagine that. There's a pandemic, people's lives at stake, and it will be discussed at a meeting a couple of months from now. Another reason the blood tragedy occurred beyond the systemic failings is, again, similar to the issues we face today with COVID ravaging the poor and underprivileged. The title Unspeakable was motivated by the prejudice directed at high-risk groups back then. The 4-H Club, homosexuals, Haitians, heroin users, and hemophiliacs. AIDS was thought of as a gay disease. It was literally called that at first, GRID, gay-related immune deficiency. I was going to repeat myself again and say, try to imagine that happening today, but sadly, maybe it's not as difficult as it should be. Back then, a culture of silence was born out of fear. Fear of being ostracized or worse, having a bullet shot through your front window like Ryan White's family. Vital information was withheld, and the excuse that was given was that blood product users couldn't properly weigh the facts. We would panic. The experts knew what was best. People were sick and dying, and no one was talking because they were terrified. In the introduction of his book, Andre Bacard admitted he covered the story for 9 years before he realized he was being lied to. When the story finally broke, it was an awakening for Canada, ushering in a new age of skepticism unlike anything that had ever been seen before in this country. We were no longer immune to scandal, and we no longer knew who we could trust. Today, we have seen when it comes to our healthcare, it's vitally important that we know who to trust. Misinformation is everywhere and can be deadly. One of the questions I've been asked the most about Unspeakable is, what do you hope people will take away from the series? 6 months ago, I would have said I was just trying to give people some sense of what it was like to live through an epidemic. Now, Unspeakable as to where we are today. If nothing else, the show introduced me to some of the incredible heroes of the Tainted Blood story. Vic Parsons, Bill Mindell, David Page, Justice Horace Creever, Marlis Edwards, Christo Soukas, survivors like Rick Waynes, Janet Connors, and of course, those that died nobly fighting for other victims like Randy Connors, John Plater, James Krepner, and Ed Cuban, to name a few. My own story does involve some hardship, but I've also been very fortunate to have excellent hemophilia doctors and nurses like Jerry Teitel and Ann Harrington in Toronto, Shannon Jackson and Deb Gu out here in Vancouver. There's no doubt comprehensive care clinics helped me lead a better life with hemophilia, and I'm thankful for that. 51 years ago, when my parents were first told they had a hemophiliac son, they were traumatized by the prognosis. Now I'm part of a generation forging new ground. They're living longer, healthier lives than ever before. We have pegylated products, safe clotting factor, new treatments like Hemlibra, and revolutionary genetic cures finally, hopefully, on the horizon. I'm incredibly grateful to the frontline healthcare workers putting their lives on the line during this current pandemic, and all those, like many of you, working to make the future healthier and safer. As we go about our daily lives trying to keep our own little world functioning, it's tempting to want to turn away from the scariness of the big picture, or mistakenly fall into the trap of thinking we can't change things for the better. As bad as it was, the blood scandal forced the hemophilia community to fight back, to speak up and be heard. It's how change happens, but it's also critical that those with more power, those who can affect meaningful change, stop and listen. One of the most common threads I heard from those who lived through tainted blood was that back then they felt like no one cared. First and foremost, healthcare is about easing and ultimately preventing suffering. People who are suffering and those who represent them need to be heard. And with that, I'll say thank you for listening.
Thank you, Rob. I will open this up for any comments or questions or discussion. I know Jerry's speaking next, so he may want to say a couple of things. I would point out that normally, actually, I introduced the history of the Center for Blood Research with my first slide is, the tainted blood. And it's really the origins of the Center for Blood Research. I knew you were speaking, and I thought, oh, I don't really have to do that today. But it's a constant, and I do present it all the time. It's a constant reminder. I think the reminder has to be made for going forward, so we avoid these disasters in the future, and that we're always aware. And here we are in another pandemic with lack of knowledge, fear, intrinsic to the whole situation with inevitably people get targeted inappropriately or whatever through ignorance and lack of science and lack of sensitivity. So it's an important reminder. Are there any comments or questions anybody would like to to speak? It's a great, fascinating program, a series to watch. If you haven't, I'm not sure how it's available.
It's on CBC Gem, which is an app you can get on for free on all your devices and computers. And
Okay.
so yeah, it's free to watch with commercials, or I think it's $4 to join and get it commercial free.
Great. Well, if there aren't any comments, I want to thank
Thanks very much for having me.
for coming and to speak to us. It was fabulous and really interesting.
Thank you very much. I appreciate the opportunity.
Great. All right. Well, I have comments coming in. It was an excellent talk. All right. Good. Okay. So our next speaker, who's already been sort of introduced, but I will introduce them. Where are we in the schedule? Make sure we don't have a break. Yes. Okay. So Jerry Teitel, longtime friend and colleague. So Jerry Teitel is a professor of medicine at the University of Toronto, staff physician in the Division of Hematology Oncology at St. Michael's Hospital, and the medical director of the Hemophilia Treatment Center there, which is indeed the largest such program in Canada. Dr. Teitel obtained his medical degree at U of T, followed by a research fellowship at Harvard University, same place that I went to, later than he did, where he worked on novel assays for the detection of coagulation activation. He returned to Toronto, where he studied the immune accessory activity of vascular endothelial cells, really ahead of his time on that, and then focused on clinical research in the field of hemophilia and related bleeding disorders. Jerry's been the president of the Canadian Hemophilia Society in the past, the Association of Hemophilia Clinic Directors of Canada, and the Hemophilia Research Society of North America. He's remained active in all of those organizations, as well known in the hemophilia space, really around the world. So we're really pleased to have him. Jerry, are you there?
I'm here, Ed.
Good, good, good. Whew. All right. I gotta find your slides though.
Okay. I'll let you drive.
Okay, you trust me after all these years. You have no choice. I'm just going to put.
While you're getting them up, I want to thank you and the organizers for inviting me. It's a great honor to be here, and it was a real treat to see Robbie Cooper on the same program. It's been a long time.
I didn't know. Yeah,
this is a bit of an anticlimax, though, because I'm not talking about congenital hemophilia today. I'm going to talk about acquired hemophilia. And the next slide gives my disclosures. And then we'll get right into it. So I'm going to start, the terrible way to start a talk, I'm going to start with an apology for showing a coagulation activation here, or coagulation slide, because I know this puts a lot of the audience to sleep, but I promise this will be the only slide showing this reaction. And I just want to bring all of you up to date so that some of you are not coagulation aficionados like Ed and me. So I want to show you what factor VIII does for a living, and it's shown there in red. Factor VIII is a cofactor in the reaction for which factor IX, activated factor IX, is the enzyme. The substrate is factor X, and the product of the reaction, factor Xa, is necessary to generate thrombin, the ultimate enzyme in the coagulation system. The next slide, and what is acquired hemophilia? So this is a rare disease which is autoimmune, and it's caused by autoantibodies to factor VIII. Now, the incidence is stated to be in the order of 1.5 per million per year. I think that's an underestimate. We certainly see more than that in the Toronto area and in Ontario, and other colleagues in Europe and Germany have had the same experience. It is generally a disease of the elderly. The median age is in the 60s or 70s. We often see patients in their 80s or 90s, and the mortality is still fairly high in those over the age of 65. Some patients we see do have an underlying disease, whether it's autoimmune, malignant, inflammatory, skin diseases like bullous pemphigoid, or infectious diseases. But in at least half of cases, there's no clear disease association. Now, these are older people. They often have concurrent disease, but usually we can't infer causation. So the treatment of acquired hemophilia is twofold. We have to stop bleeding because the disease usually is symptomatic. It's rare to see this asymptomatically. And we like to try to eradicate the inhibitor to restore hemostasis to normal. The bleeding pattern, and these last two bullet points I've highlighted because these differ from bleeding, from the status in congenital hemophilia. Bleeding tends to be subcutaneous, intramuscular, GI tract, genital urinary, and retroperitoneal, and it correlates poorly with the factor VIII level. And the next slide shows what it looks like. And the picture on the left is a current patient of mine. There's a woman in her 90s who's currently in hospital. And the picture on the right and the accompanying scan showing intramuscular hematoma is from a published case from Jean Saint-Louis at Maisonneuve-Rosemont Hospital in Montreal. So the next slide, please, shows something about the antibodies in acquired hemophilia. We typically call these inhibitors because they inhibit function. So inhibitors can occur also, in fact, frequently occur in congenital hemophilia, specifically people with severe hemophilia, but those are alloantibodies as opposed to autoantibodies. There are alloantibodies that occur after replacement therapy, so in response to replacement therapy with factor VIII. But even these patients, congenital hemophilia patients with inhibitor antibodies, have different clinical and laboratory features from patients with acquired hemophilia. And some of those differences are shown here. You can see that the bleeding pattern is acquired hemophilia patients, as I told you, have subcutaneous and mucocutaneous bleeding, which is different from the deep bleeding we see in congenital disease. And this is an important point. The second row shows that these antibodies in acquired hemophilia are usually incompletely inhibitory, and they follow complex kinetics, and I'll show that in a subsequent slide. And that's different from inhibitors that arise in congenital hemophilia, which are usually completely inhibitory and follow first-order kinetics. In both diseases, both situations, we can use bypassing agents, which we'll touch on again a little bit later in the talk. But in terms of eradicating the inhibitor antibody, immunosuppression is more effective in acquired hemophilia, whereas in congenital hemophilia, when inhibitors arise, we generally treat these with immune tolerance induction, that is, with high doses of antigen factor VIII rather than with immunosuppression. On the next slide shows you some important questions that I'm going to address briefly during this talk. First of all, why are these clinical pictures of acquired hemophilia different from those of congenital hemophilia? In both cases, the only pathological event is factor VIII deficiency. So why do they look different? Second question, what are the most effective and safest regimens to treat bleeding in acquired hemophilia? Then, what are the most effective and safest treatment regimens to eradicate the inhibitor in acquired hemophilia? And finally, are there things that we can look at to predict the natural history and prognosis to try to rationalize our treatment and minimize toxicity? So the next slide. So the first question we'll address is, why does bleeding differ in patients with acquired hemophilia and congenital hemophilia? And the next, please. If you look at the left, this illustrates what I mentioned before about the nature of the kinetics of the inhibitor antibodies. So the curve in the red shows the typical kinetic pattern of autoantibodies in acquired hemophilia. You can see that this is a semi-log plot, and you can see that the inhibition of factor VIII, so the plot shows residual factor VIII as a function of incubation time. This shows that the kinetics are nonlinear, this is curvilinear, and incompletely inhibitory. There's residual factor VIII detectable, as there is in many but not all such cases, despite the presence of a high titer antibody. Whereas the curve in blue shows the type 1 linear kinetic inhibition of the antibodies, which typically arise in 30–35% of patients with severe congenital hemophilia. Next slide then asks this question, or tries to approach an answer to the question, first of all, why does hemophilia cause bleeding at all? Sounds like a pretty straightforward question. So if you look at congenital versus acquired hemophilia, in both situations, we have sites of tissue factor concentration which is insufficient to generate enough thrombin to form efficient clot via the extrinsic pathways. You know, tissue factor is the trigger for extrinsic coagulation. And in both situations, we have factor VIII deficiency, whether it's on a congenital basis or acquired due to an autoantibody. But if you look at the left-hand box, the third bullet point shows an interesting observation, which was made now many years ago by investigators in Europe, and also by the late Dr. Nesheim in Queen's University, Mike Nesheim. And this is the observation that the impaired thrombin generation in hemophilia leads to reduced activation of TAFI. TAFI is the thrombin activatable fibrinolytic inhibitor, a major player in fibrinolysis. And the result is that bleeding in congenital hemophilia is not only caused by hypocoagulation, it's also caused by unregulated fibrinolysis. So bleeding is caused by both coagulation insufficiency and excessive fibrinolysis. Whereas in acquired hemophilia, due to the complex kinetics of the inhibitor antibody, there is typically enough residual factor VIII to generate enough thrombin to activate TAFI. So bleeding in acquired hemophilia is caused by impaired coagulation, but fibrinolysis remains normally regulated. And this, I think, is a clue to why the bleeding patterns differ. Now, the next slide asks another related question, and that is, what about site specificity? Why do we see subcutaneous and mucosal bleeding in acquired hemophilia different from the deep bleeding in the congenital disease? Hemophilic bleeding, congenital hemophilic bleeding, occurs predominantly at sites where there is limited tissue factor density, where there will be, as I said, deficient TAFI activation and excessive urokinase-mediated fibrinolysis. And this is the typical situation in synovial membranes, which is why the major site of bleeding in congenital hemophilia is intra-articular. On the other hand, in acquired hemophilia, incomplete inhibition of factor VIII activity by antibodies with the type 2 kinetics that I showed you allows enough thrombin generation in some sites, such as joints, to activate TAFI, thus regulating fibrinolysis. And a little side note here in the asterisk is that there are some rare situations, these are quite uncommon, in people with non-severe hemophilia, mild and moderate hemophilia, who do develop alloantibodies, inhibitors. And in these cases, many of these patients bleed not like those with severe hemophilia, but like patients with acquired hemophilia. In other words, they show the same type of type 2 kinetics that we see in acquired hemophilia. The next slide, we're now going to approach the question, what about current treatment approaches to treat bleeding? And the next. So hemostatic therapy in acquired hemophilia. First of all, just to get rid of these very quickly, desmopressin, DDAVP, and human factor VIII concentrates have very limited roles. In a very few patients who have very low titer inhibitor antibodies, one might be able to use them, but it's very rare that they would have any roles. So the traditional approaches to achieving hemostasis involve inhibitor bypassing agents. That is, agents which promote hemostasis distal to the block imposed by the inhibitor antibody. And the typical, or the traditional inhibitor bypassing agents we use are activated prothrombin complex concentrate and recombinant activated factor VII. And there's only one commercially available source of either of these products. They've been around now for a long time. They're quite efficacious in the 80 to over 95% range in registries that we have available. However, their efficacy is unpredictable, and we don't have validated laboratory monitoring to be able to monitor their efficacy. The other approach, which was traditionally used and is no longer available to us, was the use of a plasma-derived, incompletely cross-reactive animal-derived factor VIII. And it so happens that porcine factor VIII is very effective as a cofactor in human plasma. And so for many years, we used plasma-derived porcine factor VIII concentrate to treat people with both acquired and congenital hemophilia with inhibitors. Unfortunately, that product had to be withdrawn in the early 2000s, and it was withdrawn because it was found to contain porcine parvoviruses. Now, porcine parvoviruses are not pathogenic in humans, but because they were there, the regulators did not allow the product to continue to be sold. And no matter what the manufacturer did, they could not get rid of those parvoviruses. They're heat resistant, they're difficult to filter, and so the product was withdrawn. So that led to the next slide, please. And that is a more recent option, which is recombinant, a recombinant version of porcine factor VIII. So the rationale here is really twofold. First of all, replacement therapy is more predictably reliable than bypassing therapy. And recombinant porcine factor VIII has, as I said, full hemostatic activity, but only limited immunological cross-reactivity with human factor VIII. And we had the legacy of successful use of the plasma-derived version up until 2004. So the product we have now is a recombinant B-domain deleted porcine factor VIII complex called Sasoctocog is the generic name. And it is approved and available in Canada for treatment of acquired hemophilia. Some of the advantages are that we can use standard factor VIII assays. So in fact, it's best assayed using a one-stage PTT-based assay and not chromogenic factor assays. And these are available in virtually all coagulation labs at coagulation centers. But it's also desirable to have an anti-porcine inhibitor assay on site to be able to monitor the degree of cross-reactivity of the anti-human antibody, as well as the formation of de novo anti-porcine antibodies, which sometimes complicate its use. So the next slide shows some new hemostatic approaches that might potentially have a role in acquired hemophilia in the future. So I'll emphasize that these are not approved uses. In fact, these products are not yet available. They're not yet licensed for use. So the first category of products are those used to treat for bleeding prophylaxis, not for treatment, but for prevention of bleeding in patients who have not yet achieved a remission from acquired hemophilia or who have been refractory to immunosuppression. And the first product is something that I think many of you will be aware of and may have used yourselves, and that is emicizumab, which enhances procoagulant activity by functioning as a surrogate for factor VIII. It is a bispecific antibody which has specificity for both the enzyme and substrate, that is factor IXA and factor X, for which factor VIII is a cofactor. Now this product is available. We are using it in Canada, but it is not indicated in acquired hemophilia, so we can't use it in this disease. Or I should at least say not yet labeled for use in acquired hemophilia. The other approach is to downregulate the endogenous anticoagulant activity. And the two targets, the two endogenous anticoagulants that are being targeted are TFPI and antithrombin. TFPI, the tissue factor pathway inhibitor, is the major regulator of extrinsic coagulation. And the approaches used to target this are monoclonal antibodies. And the one which is farthest in development in phase 3 clinical trials is called concizumab, but there are others. And the approach to downregulate antithrombin, or AT3, is a molecule called fitusiran, which is a small interfering RNA, which reduces transcription of the RNA, of antithrombin. So these are both in phase 3 clinical trials, and perhaps at some point they may have a role in selected patients with acquired hemophilia. There are risks and uncertainties. Anytime we use products such as this, there is a thromboembolic risk. Particularly, remember, we're dealing with very old patients generally. And this risk is especially high when used in combination with bypassing agents. And we will need bypassing agents or recombinant porcine factor VIII in patients who do have breakthrough bleeding despite prophylaxis with the agents that I just mentioned. And finally, it's important to remember that monitoring is always top of mind for us, and hemostatic monitoring may be affected. For example, some of you will be aware of the fact that any PTT-based assay, such as the PTT itself or a factor VIII one-stage assay, will be invalidated in the presence of emicizumab. So the next slide. We'll now look at the question of eradication of the antibody by immunosuppression. So the next slide I'm showing this, this is work done back in 1993, published by David Green and colleagues in Chicago. And I'm showing this because as far as I know, this is the only randomized and it's tough to do randomized controlled trials in rare life-threatening diseases. And this trial didn't really tell us very much. The numbers were small, But what they did is treated patients up front with prednisone for immunosuppression, and in those who did not have remission with prednisone, they randomized them to either steroid alone, cyclophosphamide, or the combination. So the next slide shows that since it's difficult to do these trials, we rely on things like meta-analyses and registries. And these were two very nicely done meta-analyses that were published in fairly recent years. And I've just pulled from them some of the data looking at this question of the relative efficacy of corticosteroids alone versus corticosteroids plus cyclophosphamide. And you can see that in the two meta-analyses that I've shown here, both of them show better results with combined therapy. So the complete remission rate in the two studies, 89% versus 70% for combined therapy, 82 versus 68%. Overall mortality is higher in the steroid-only group, 26 versus 16 and 24 versus 20. And in the analysis, which also looked at disease-specific mortality, again, higher mortality attributable to corticosteroids alone. The next slide shows one of the registries, and this is a registry which is, has been a real important source of information to us, published recently. This is called the EACH2 registry. This is the European Acquired Hemophilia Registry. And I'm just pulling some of the data from one of their papers, which again looks at this question of the relative efficacy of steroids alone versus steroids plus the cytotoxic drug cyclophosphamide. And again, similar results and pretty good numbers here, 142 and 83. And you can see that the complete remission rate, 80 versus 58% for combined therapy, higher rate of stable complete remission, so a more robust response, 70 versus 48%, and a lower relapse rate. Now, cyclophosphamide is a toxic drug in some of these elderly patients, so we can't always use it or we can't always complete the course, but where it's available or where we can use it successfully, it does have an important role. The next slide, what about the question of our favorite immunosuppressive drug these days, and that is rituximab, anti-CD20 monoclonal antibody. So the first case reports go back to 2001, and subsequently there have been a number of reports of the use of rituximab as either first-line or salvage therapy, either alone or combined with steroids and cyclophosphamide. And again, the EACH2 registry showed some data which don't look very impressive when you look at these numbers. 59% response rate given rituximab containing regimens up front to eradicate the inhibitor. Small number of patients given rituximab alone had a 42% response rate with a high rate of adverse events, and an overall response rate of 64% for all rituximab containing regimens. Now, I think one of the reasons these numbers don't look very impressive is that rituximab has often been used as a sort of desperation therapy in patients who have failed first-line therapies. And indeed, we typically reserve rituximab now as second-line therapy for patients who have not responded adequately to first-line therapy or in whom we believe the prognosis may be very poor. Now, the next slide, we'll now look at the last few slides to ask about natural history and prognosis. Is there some way we can identify patients who are at higher risk so we can perhaps treat them more aggressively up front? And the next slide shows several surveys which I have put together on this figure to look at deaths in mortality in acquired hemophilia. So there's an international survey, a UK, a French study, EACH2 registry, and another very important study called the GTH registry, which is a German and Austrian study. And if you just look at the bottom two rows that I've highlighted, the overall deaths you can see are still quite high, ranging from 26 to 41%, but most of these deaths are not due to bleeding. So in other words, most of these patients die either from underlying disease, remember they're generally very elderly, or importantly, from the toxicity of our treatments, primarily from infections related to our immunosuppression, so iatrogenic deaths. In the next slide, what about predictability? This is data from the GTH registry that I showed. That was one of the registries shown on the previous slide. And the first two plots, these are Kaplan-Meier survival curves, are perhaps not too surprising. These show that the presence of malignancy and poor performance status are adverse prognostic signs with respect to death. But the figure on the right is perhaps a little more interesting and not as predictable, and that is that patients whose factor VIII is undetectable at diagnosis have a poorer prognosis than those who have residual detectable factor VIII. So that perhaps allows us to identify a group of patients for whom upfront treatment can be more aggressive. And the next slide is also from the GTH registry, and this is quite interesting. This is something we don't usually do clinically, but something that we're going to have to start thinking about. And that is, if you look at the heavy chain isotype of the immunoglobulin, the inhibitor antibody itself. And this shows that patients who have anti-factor VIII IgA have a worse outcome in acquired hemophilia. So the left-hand figure shows complete response rates. The middle, you see they have a higher recurrence rate, very poor robustness of the response in the presence of IgA antibodies, and even a tendency towards poorer survival. So we're approaching the ability to understand which patients are going to do worse up front, and perhaps this will help us to inform treatment. So finally, the next slide, those of you who are interested perhaps in getting a little more deeply into this topic, we can't publish good guidelines on acquired hemophilia because they're not the high quality guidelines that we would like to have. So the best we can do, and this is an international group we put together to issue recommendations, and that's the only word we could use is recommendations rather than guidelines on the diagnosis of and treatment of acquired hemophilia, and this appeared recently in the journal Hematologica. So with that, in the last slide, I will thank you for your attention. And you saw this picture at the beginning of this symposium. It's not Ed Conway's summer home. It's the birthplace of Norman Bethune. And it was just coincidental that Ed and I picked the same picture to show, but if this illustrates that I'm starting to think like Ed Conway, I will take that as a high compliment. Thank you.
It's the reverse, but thanks. Thanks, Jerry. That was fabulous. Whoops. So let me stop this here. Okay, and get that out of that. And we can open this up for questions or comments. Anybody, we have some hemophilia docs in the crowd, I saw. So while we're in case, in the meantime, I will ask. So the porcine, the recombinant porcine factor VIII. So I knew it was coming. I'm not in the field, so I didn't know how much it's used. Are there antibodies raised against the porcine factor VIII?
Yes, there can be. I'm surprised that they occur as infrequently as they do. When they do, it's usually not before at least 7 to 10 days, usually longer. And by then, you've usually got control of hemostasis. So, you use it on a rainy day, it's raining, you use it, and then you develop the antibody. But the majority of our patients, and we're using it as first-line therapy now in most of our acquired hemophilia patients, we have only infrequently seen it in patients who didn't have any cross-reactivity at baseline. So it's either not that immunogenic, or our patients, of course, are immunosuppressed because we're treating them with steroids and sometimes cytotoxic drugs. So it's not a major problem. It's a bigger problem if you've got pre-existing cross-reactivity, which prevents its use up front. Even then, though, you can often use higher doses and get good levels. And the pivotal study published by Rebecca Cruz-Jarras and colleagues showed that nicely.
We have a few questions. Ed Prysdale has arrived because he just was flying in. What is the frequency of IgA and acquired factor? He actually wrote 7, but I'm sure he meant factor VIII deficiency. Ed, you should be sleeping.
Sorry, the incidence?
The frequency of IgA.
The frequency.
Yeah.
You know, the only publication that addresses this was this GTH survey, and it was a minority, and I don't remember the numbers. It was sufficient enough that they were able to break it out and show an effect on survival. But you know, no one that I know of is doing this routinely yet. But it's something that we'll probably we will probably consider starting to do. But I can't answer that question directly.
Okay, so we have, how would you, I guess, organize in terms of order of choice, the different treatments for controlling AH bleeds now, recombinant porcine factor 8, APCC, recombinant 7A, what you'd expect for future molecules.
Right. Well, as far as priority or my my choice of treatments, where it's possible to use porcine factor 8, we are considering this. My colleague Michelle Scholzberg and I are considering this to be first-line treatment. And the advantages are, first of all, it's more satisfying and more logical to use authentic replacement therapy rather than bypassing a deficiency. And you can measure it. You measure it using the same assays. You know, if you're going to get a normal factor 8 level, you know you're going to get hemostasis. With factor 7A and APCC, you kind of cross your fingers and hope it's going to work. Now, it usually does, but not always. So there still is a place for the bypassing agents. We'll often use factor 7A for early control of a bleed. I think it works better when used early rather than late. Works better for mucosal bleeds than for deep bleeds. And it's often good during surgery. It becomes a little cumbersome after surgery because it has to be given very frequently, very short. It's got a short half-life for a drug, a long half-life for an enzyme, but short for a drug. And APCC is very effective as well and can be given less frequently. So we really use them both with good confidence, and we've been using them for many years. But if we can replace with a non-human factor 8, we prefer to do that.
Christian, you want to unmute?
Thanks. Yeah, I was interested in the differences in fibrinolysis between congenital and acquired. And I was wondering about antifibrinolytics. Is tranexamic acid, is that used differently for congenital and acquired?
Yes, first of all, for mucosal bleeding and congenital hemophilia, tranexamic acid has been used for many years. So we often use that to supplement replacement therapy. One exception is urinary tract bleeding, because sometimes it will cause clotting in the urinary tract, and we end up getting obstruction from clot. But otherwise, for mucosal bleeding, nasal mucosa, oral mucosa, uterine bleeding, it's very effective when used together. In acquired hemophilia, rather, less often. We will often use it with factor 7A. We're more reluctant to use it with APCC only because the label discourages us from doing that, but also because these patients, as I have said several times, tend to be very old, and we don't want to over-coagulate these people who may be a priori at higher risk for thrombosis. But it does have a role, particularly for mucosal bleeding in congenital hemophilia, a big role, yes.
Question from Shannon. Hi, Jerry. Have you ever had to use bortezomib or daratumumab in the setting of refractory AHA? And maybe you'll first explain what those are.
No, so these are monoclonal antibodies. These are more these are drugs that are approved for use in lymphoproliferative diseases, not in acquired hemophilia. I haven't, Shannon. I have used mycophenolate. Got a patient on mycophenolate now, and I've used cyclosporin, but I have not gone to any of these newer agents. They're certainly not approved. There are perhaps case reports, and Shannon's asking it, so I wonder if she may have had some experience with it herself.
Shannon, have you used it? You can unmic. You can open the mic.
She's saying no. Okay.
Okay. Nope. Sadly. All right. Any other questions or comments?
Yeah. Yeah, I have one. It's Prysdale again. I missed, very sorry that I missed the first 2/3 of your talk. So you might have already addressed this, but I was curious if we know anything more about antibodies to emicizumab and whether or not these antibodies are messing up that drug.
Yeah, as far as I know, Ed, these are very rare. They have been reported, but very rare. We're using it now routinely in congenital hemophilia with inhibitors in Canada, and in the United States, it's used even for congenital hemophilia patients without inhibitors. And it has not been a problem. I've not seen reports of that being a problem, so it's quite rare. And by the way, emicizumab is approved for non-inhibitor congenital hemophilia patients in Canada, but it's not being made available yet through the CBS, pending a CADITH review.
Thank you.
We do have one more question. Okay, Nick Sunderland, Obizur, any issues practically? I'm not sure. We're given multiple violets and not stocked in BC. Should things be moving?
Okay. Yeah, I think I get that. Obizur is recombinant porcine factor 8. And one of the points is a very practical one that he's making. I believe it's available only in 500 unit vials. And those are very small because typically a dose might be in the order of 50 to 100 units per kilogram. So it's a big burden on the blood bank staff to prepare this stuff. You know, that's invisible to me as a treater. Of course, I just have to write the orders. So, so it is cumbersome in that sense. But and we have not had a problem with availability, certainly in Toronto or in Ontario. But I guess from the question, you're indicating that it may be, you may have a little bit more problem accessing it in BC. Now, you know, it's approved through through CBS and through EMA Quebec, so it's approved and it's on the inventory. So you know, I guess a center that uses it a lot, such as us, we have not had problems accessing it.
Okay, so I think we'll wrap that up. We're right on time. Jerry, thank you so much. And I know that everybody enjoyed it, and we had amazing kudos from Rob Cooper as well. Obviously well earned, and we knew that though. So thanks very much for that great talk.